Hello everyone and welcome to today's live broadcast, Battle the Heat in Your Lab, Facing Distorted Results Caused by Temperature Fluctuations, presented by Dr. Gediminas Vidugiris, a Senior Automation Scientist at Promega Corp, and Dr. Katrin Flatscher, a Marketing Application Specialist Detection at Tecan Austria. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Tecan. In this webinar, participants will discover the benefits of active temperature control supported by experimental data from cell-based bioluminescent and biofluorescent assays assessing cell viability, metabolism, death, and signaling. You will learn how to design assay and signal measurements to generate reliable and reproducible data and avoid false positives. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Q&A button located at the lower left of the presentation window and type your questions into the box that appear on the screen. We'll answer as many questions as we have time for at the end of this presentation. Also, please notice that you are viewing this presentation in a slide window. To enlarge that window, just click on the screen icon located at the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window or use that Q&A button and let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Click on that button at the bottom left corner and follow the process for obtaining your credits. I'd now like to introduce our two fabulous presenters. First, Dr. Gediminas Vidugiris. Dr. Vidugiris is a Senior Automation Scientist at Promega Corp in Madison. He received his PhD in Biochemistry at the Institute of Biochemistry, Lithuania Academy of Science, Vilnius, Lithuania. Dr. Vidugiris has extensive multidisciplinary research and development experience in systems automation, screening, and biodevelopment, biochemistry, biophysics, spectroscopy, and electrical chemistry. And our second presenter is Dr. Katrin Flatscher. Dr. Flatscher is an application scientist at Tecan Austria. She studied molecular biology at the University of Salzburg, focusing on cell biology and immunology research. I'd now like to welcome both presenters. Welcome. And we'll start with Dr. Vidugiris.
Thank you, and hello, and thank you for joining us today. One of my duties at Promega is to evaluate and validate newly developed Promega assays by screening them against small compound libraries. After processing multiple screening data sets, I was quite puzzled by the jigsaw pattern or periodicity of the data I observed in all of my screens. The graph on the left data of 16 plates shows for each plate displayed an obvious gradual signal increase through each of 80 data points. We'll come back to this graph a bit later. Of course, much more desirable and expected data distribution would be on the right graph. This observation inspired me to investigate this phenomenon and to find the solutions to avoid it. We know that all chemical reactions are affected by the temperature. This effect is quite significant. Reaction rate acceleration of 2 to 3-fold is observed with temperature increase for 10 degrees of Celsius, as described by Arrhenius equation. All instruments generate heat, which affects the chemical reactions monitored inside of those instruments. The Tecan T-Cool module is capable of maintaining stable temperatures from 18 to 42 degrees Celsius, which mitigates this issue. Today I will present benefits of multimode reader equipped with T-Cool an active cooling module versus readers which are not actively cooled. This investigation employed multiple bioluminescence and biofluorescence reactions. Multi-well plate read time inside the reader depends on a couple of factors: number of wells per plate and also signal acquisition time per well. As you see on the graph, the read time of the higher density plates is quite significant. The plate residence time inside the reader is sufficient to heat up the plate from 2 to 10 degrees Celsius depending on the reader. All the measurements I present today were assembled in the 384 well plates with the acquisition time of 300 milliseconds per well. Based on these settings, the read time for one plate was just under 3 minutes. Assays were set up in low volume 384 well plates. Total reaction volumes were 18 microliters. To investigate the reaction volume temperature increase inside the reader, I investigated situations where entire plate was filled with the same reagents, while in the other plates plate was split to vary one of the reaction reagents components, as shown in the picture. All data points obtained from the wells filled with the same reagents were averaged, and this average data value provided one data point for the assay signal kinetics observation graph. Two separate plates were filled with the same reagents and monitored simultaneously in two separate readers. One plate was placed in the T-cool spark reader and the other plate in not actively cooled reader. For each plate, reads were taken every 5 minutes, and the signal across the plate were averaged to generate one data point per read. In this graph, we can see how overall reaction signals changes over time in the reader. You will see these types of graphs through the presentation. On the left is the raw signal data graph, and on the right is normalized by the maximum signal graph. Now let's start with the example of bioluminescence reaction signal variations. This is bioluminescence Succinate-Glo assay, for which temperature has one of the largest effects on the reaction signal. Data points indicated by the filled circles were obtained on the not cooled reader, and data points indicated by the open circles were obtained on the T-cool spark reader. As you see on the T-cool reader, signal increases continuously for the first 20 minutes and then starts decreasing. Contrast this with the Tecan cool reader where signal measurements immediately starts gradually decreasing. The signal strength for the first measurements is quite similar between both instruments. On the left graph, you see it's around 150 and probably like 30,000 counts. Measurements were obtained at two substrate concentrations. While the magnitude of the signal for the two substrate concentrations differ significantly, the normalized graph shows that Succinate-Glo assay rate is concentration independent. The succinate consumption rate is equal at both succinate concentrations 1 and 5 micromoles, light brown and dark brown curves. What that affects the reaction rate, it's not the substrate concentration, but the temperature. After initial increase in the reaction rate, the reaction signal in the not cooled reader clearly decreases at a faster rate than those in the actively cooled reader, right graph. Biofluorescence reactions also are affected by the temperature. One of the Promega's cell viability assay is Assay-Glo, which evaluates cell viability biofluorescence signal. Assay-Glo assays cell-permeable glycine-phenylalanine AF coumarin substrate enters the cell, where it is cleaved by the live cell's protease activity to produce the fluorophore AFC coumarin, or AFC fluorophore. Excitation for this fluorophore is 415 nanometers and emission maximum is 500 nanometers. Production of AFC compound in healthy cells is reflected in the graph on the right. When measured on the not cooled reader, the reaction volumes warms up and the reaction rate increases, producing more AFC fluorophore green and yellow lines. Operating at the room temperature in the T-cool spark, AFC fluorophore accumulation is slower and signal increases more slowly, blue line. In reactions with anti-cancer drug Velocade and the K562 cells, lower left graph, protease in apoptotic cells becomes inactive and AFC production is significantly slower, producing significantly lower fluorescence signal. In an actively cooled T-cool reader, the reaction rate is slower and fluorescence signal is slower in comparison to the signal obtained on the not cooled reader. In the biofluorescence reaction, signal continuously rises, like as you see here, because more and more fluorophore is produced and it accumulates in the reaction volume. Contrary to bioluminescence reactions, where substrate is depleted by reactions, causing the luminescence signal to decrease. This slide highlights how sensitive the reaction signal is to even small temperature variations. We are looking at luminescence kinase glow signal over time. Shown in the red is the curve produced by a not cooled reader operating at 35 degrees Celsius internal temperature. Temperature on the T-cool spark was adjusted in small increments and kinase glow assay signal measured repeatedly, with each temperature increase of 1.5 degrees Celsius, signal intensity increases. The normalized graph on the right shows how much faster the reaction substrate consumption is on the subsequent signal decrease on the not cooled reader, red curve. Different bioluminescence assays are affected to different degrees by the temperature. In some bioluminescence assays, for example, Succinate-Glo assay, the signal increase caused by the temperature is small, only about 5%. The largest signal increase of about 20% was observed, for example, UDP-Glo assay, and the largest, up to 40% signal increase effect caused by the temperature was observed for Succinate-Glo and ADP-Glo assays. This observed time effect on the signal prompted assay developers to introduce the pre-read incubation times of 10 to 15 minutes in their assays. This was presumably to allow luminescence signals stabilize before measurements. But as my work indicates, this incubation time is not necessary and may increase overall variability of the assay signal. This slide shows bioluminescence signal changes over time as blue curves, and the signal percent CV, or coefficient on signal variation, changes in time as brown dash curves. Scale from those CV curves are on the right-hand side of the graphs. On the left slide side, data are displayed from the different assays obtained on the not cooled reader, and on the right slide side, data for the T-cool spark. In all graphs, luminescence data behave as we discussed earlier. For the not cooled reader, we observe signal increase associated with reaction temperature increase and luminescence increase, as absent for the data obtained on the T-cool reader. But %CV or signal coefficient of signal variation, brown dash curves, is increasing with time in all the graphs. This signal variation increase combined with the overall signal decrease can significantly negatively affect assay Z prime values. Top 4 graphs in the top row illustrates the misconception that waiting 10 or 15 minutes will allow luminescent signal to stabilize. As you see here, it's no difference if you dispense and measure or dispense, incubate 10 minutes, and then measure luminescent signal. For the not cooled instrument, 2 top left graphs, luminescent signal increases independently whether you wait 10 minutes or not. And for the T-cool spark, luminescent signal continuously decreases independent of 10 minutes incubation. So again, I want to underline here that assay can be and should be read right after dispensing and mixing, of course. This can also significantly reduce the processing time for multiple plates. But there is an exception when complicated multi-step bioluminescence reactions are taking place. Then the much longer incubation times of half an hour, 1 hour, or 1.5 hours are required for the assays, and those incubation times should be upheld. Now coming back from where I started, the idea of an internally cool reader, and this work was instigated by my small compound library screening results. LOPAC, it's a library collection of the 1,280 pharmaceutically active compounds provided by Sigma Aldrich. This library is supplied in 16 racks with 80 compounds per rack. So 16 racks multiplied by 80 compounds is equal with 1,280 compounds. LOPAC compounds for screening were dispensed 2 nanoliters per well by lab-side echo-acoustic dispenser into the 384 well plates. I screened the 80 compounds of the supplied in the rack in quadruplicates in low 384 well plates. So 80 compounds multiplied by 4 or quadruplicates is equal to 320 wells, and I had still 64 wells left for controls. To screen all 1,280 compounds in the LOPAC library required 16 384 well plates. Plate numbers are in the green ink at the bottom of the graph. After screening quadruplicates, results for each compound are averaged, and each average value is represented by a single data point on the graph above. And if you look at the results from the screens, you see jigsaw pattern or periodicity for each plate or every 80 data points. Now let's look at CMP-Glo assay versus LOPAC screen data. On the left graph, data were collected on the not cooled reader, and on the right graph, results were obtained on the T-cool spark reader. As you see, T-cool results appear significantly smoother. By calculating Z prime values for each plate, you can see the signal difference between not cooled and actively cooled readers. The lower bar chart shows Z prime values for each of 16 plates. Reads on the T-cool instrument are shown as blue bars, and reads on the not cooled reader are shown as brown bars. All blue bars are significantly higher. The average Z prime values for the 16 plates obtained on the T-cool equipped instrument, blue dash line, is 0.1 unit higher compared to the average Z prime values for a not cooled reader, light brown dash line. Z prime calculations for Succinate-Glo assay reveal even larger difference between Z prime values obtained on cooled and not cooled instruments. Z prime values average over 16 plates for a T-cool reader are improved by 0.24 units. It's a big difference. On both top graphs, the red dash line indicates 3 times standard deviation line, which marks limit below which compounds can be selected as a hits or active compounds. As you see on the not cooled reader, on the left graph, we can count just 5 compounds appearing below 3 standard deviations line. As on the T-cool instrument data, we can identify by the same assay 24 active compounds. On this slide, I show correlations graphs of the data obtained on cooled versus not cooled reader for 2 assays. For both assays, green rectangles mark higher activity points obtained on the not cooled reader caused by the temperature equilibration effect. The greater spread of the data obtained on the not cooled reader significantly impacts the ability to distinguish the real hits. Now, the adverse effects of the reaction volume heating on signal variability can be successfully mitigated by pre-warming plates before taking the measurements. Pre-warming can be done inside the reader, especially if just one or couple plates are being measured. But pre-warming also can be performed inside the dedicated incubator set to the temperatures matching the reader's internal operating temperature. All data on this slide represents measurements on not cooled reader, the same GTPase Glo assay. Top left graph shows data obtained from the plates measured at ambient temperature, and the top right graph shows data obtained with the plates pre-warming in dedicated incubator for 10 minutes each. Clearly, top left graph screen results without pre-warming plates are already very good. The Z average of the plates were 0.87, indicated very successful screen and robust assay. But pre-warming of the plates even further increases the Z prime average up to 0.93. Lower graph illustrates the benefits of pre-warming plates as evaluated by Z prime calculations for all plates with and without pre-warming plates. Pre-warming assay plate raises Z prime average by 0.06 units. So as you see, plate pre-warming can successfully mitigate results scattering caused by reaction temperature increase inside the reader. Unfortunately, plate pre-warming is also time and resources consuming step. So, in this presentation, I showed that bioluminescence and biofluorescence reaction rates are affected by the reader's internal temperature. Even small temperature changes have significant effect on the reaction rate. Different biochemical reactions are affected by the temperature by different degrees. After reagent addition and mixing, incubation time of 10 to 15 minutes, usually for most reactions, is not necessary and might be detrimental. Reading plates just after dispensing without the pre-incubation results in significant time savings during multi-plate processing. Pre-warming plates solve problem caused by the plate heating inside the reader, but it also consumes time and resources. Thank you for your attention.
Thank you so much, Dr. Vidugiris, for that important research. I'd now like to turn our presentation to the second part. Dr. Katrin Flatscher, welcome.
Thank you, Susie, and thank you, Gediminas, for this interesting summary of your measurements. On the next couple of slides, I would like to give you a brief overview how active cooling inside a multimode reader can be accomplished. The reader that was used for the studies described in the previous slides, and in fact the only multimode reader currently on the market that possesses an integrated cooling function, is the Spark reader by Tecan. The Spark is a multi-modular reader platform that combines a number of different functions and readout modes for all kinds of research objectives and application requirements. It is a spectrometer, it is an ELISA reader, it is a dual channel dispenser, it can be used as a luminometer, as a cell counter, and even as a semi-automated microscope, all in one box. As you can see, the Spark unites many different functions and modules. It offers all the most popular detection modes like absorbance, fluorescence, luminescence, alpha screen, fluorescence polarization, etc., etc. And it also comes with many available accessories like reagent injectors, a tool to prevent fluid evaporation, and of course with a very useful and easy to use software that is called Spark Control. In the context of this webinar, the functions that we would like to focus on are the integrated heating and cooling functions of the instrument. The cooling function specifically is called the T-Cool module. So first of all, the Spark instrument is able to heat up the measurement chamber from 3 degrees Celsius over ambient up to 42 degrees Celsius. This is especially useful for cell-based applications, mostly with eukaryotic cells that require a temperature that resembles more or less the physiological body temperature, so 37 degrees for optimal growth and proliferation. The software of the reader enables simple and straightforward definition of a target temperature which will be reached within minutes and maintained over days if needed, with a uniformity across the plate of 99.5%, so a results variation of less than 0.5% across an entire plate. The temperature function is indeed part of the basic configuration of every Spark reader, so every instrument will be equipped with this heating function, and it can of course be combined with various other functions designed for cell-based assays. Not only for cell-based assays, but this is, in my experience, the most popular application field for these so-called environmental control features. So researchers typically want to do long-term cell growth studies, and in this context, the heating function and the ability of the instrument to maintain a target temperature of 37 degrees is most useful. But it's not only a heating function that the Spark possesses, it's in fact the first reader on the market that not only has the ability to heat the measurement chamber, but also permits lowering the operation temperature below ambient. The minimum temperature that can be reached with this system at the moment is 18 degrees, so that's well below the normal room temperature level. It may not actually be like refrigerator conditions, but it meets the requirements of many temperature-sensitive probes and samples. And in addition, it helps to maintain a constant temperature during long-term kinetic measurements by actively preventing instrument heating. So how is this cooling function accomplished? The Spark reader, when equipped with the so-called T-Cool module, is a two-component system. It uses a special external and internal part. The external water cooling unit chills the water outside of the instrument, and this is then used to cool or as a cooling medium, which is pumped back into the reader's internal cooling module to cool the air inside the reader. And the air that has been warmed up during operation is pumped back to the cooling module, to the external part, cooled again, and then returned to the reader. And this cycle serves to maintain a specified temperature, if needed below ambient temperature, even for long-term measurement periods, so even for several days, if needed. So why is a constant, why is a constant assay temperature so important? Well, first of all, scientific studies done in different labs in different parts of the world should still have the same outcome if they're performed in the same way. A scientist in Stockholm, Sweden, should be able to reproduce the results of another scientist in San Diego, California, when using the same samples, same assay chemistry, and the same experimental setup. The components of the experiment should react and behave in the same way, no matter in which climate conditions the assay is performed. In reality, this is hardly ever the case. Reagents and other assay components, particularly enzymes and other temperature-sensitive components, have temperature optima, so that means best working conditions. And they may lose a significant portion of their activity when used outside this optimal operation temperature or the target temperature range. So for reproducible and comparable results, it is vital to maintain a defined target temperature at which the assay is performed day by day. In addition, the reaction kinetics shouldn't be influenced by temperature increases in the lab throughout the course of the day. So we all know in the mornings it tends to be a little cooler than at noon, and in the afternoon, especially during these long summer days, hot summer days, the temperature in the lab can really rise significantly. And all of this shouldn't influence the dynamics of your assays. So using any type of temperature stabilizing function like the T-Cool module will make your results much more reliable and reproducible. One of the first scientists to find out about the susceptibility of chemical and biological reactions to temperature was Jacobus Henricus van 't Hoff, a Dutch physical chemist and the winner of the first Nobel Prize in Chemistry. His pioneering work helped found the modern theory of chemical affinity, equilibrium, chemical kinetics, and chemical thermodynamics. He stipulated that enzymatic activity is directly dependent on temperature, and the corresponding equation that is still the basis for these findings is named after him, the so-called van 't Hoff equation. He found that, for example, the speed of a chemical reaction is increased by up to 12% if the temperature is increased by only 1 degree Celsius, which can lead to a variation in the final measurement results of even 10 to 20%. So really a significant change just based on fluctuations in the temperature level. And his findings can be easily confirmed in many modern day scientific applications. As we can see here on this slide, the signal uniformity of replicate samples can drastically vary when an assay is performed at different temperature levels. The green line is how it should be, you could say. A constant temperature throughout the entire measurement period means lowest possible CV, lowest possible results variation. So this is what every researcher should aim for, providing a constant temperature for the assay setup throughout the entire measurement period. In contrast, if the temperature increased by 4 or 8%, as was the case in this example experiment here, you can see a massive results change, so a massive increase in the signal itself, but also a massive increase in the CV, the coefficient of variation of the replicate samples. Particularly, the reaction suffers from the temperature increase during the readout, which, as you can see, leads to an accelerating slope of the signal curve that becomes more profound the higher the temperature gets. So this is exactly what the T-cool function of the spark reader is intended for, maintaining a constant temperature even if the ambient temperature in the lab rises throughout the analysis period. And in addition, having the option to control temperature inside your reader even below ambient opens up possibilities for new applications. For example, you can mimic the water temperature conditions for marine organisms, which usually like a surrounding temperature of 18 to 20 degrees. So that makes it an ideal tool to study the responses of marine species to climate changes, for example. And in addition, many bacterial cell cultures have a growth optimum around 20 degrees or lower. These applications can also benefit from having a cooled reader, which, for example, can be used to simulate growth conditions in the food industry or in agriculture, where researchers are interested in finding out which temperature conditions are critical for microbial growth. So just to name a few examples where cooled detection instrument can be beneficial for best possible results. At the moment, we at Tecan have some ongoing cooperations about these topics and about new application fields where actively cooled microplate readers can be helpful. So I expect I will be able to tell you more about these applications in one of our next webinars, hopefully. So to summarize these findings and the information on the last couple of slides, and to give you an overview of the most important and useful advantages of active cooling in a reader. With an active cooling system like the T-cool module for the spark reader, you can always measure under defined temperature conditions, no matter what time of day or year, independently of the location of your lab. You can perform assays below room temperature without having to work in a cooled laboratory. You can decrease the results variation or the coefficient of variation of your measurements by avoiding the instrument from heating up during the measurement. You can study new species with special growth or metabolic requirements, for example, bacteria, insect cells, or marine organisms. And lastly, you can speed up the cool down times after experiments at elevated temperature, which is especially useful if many research groups have to share one and the same instruments. So all of these benefits make the T-cool function of the spark reader very much suited for modern day research. So with this, I would like to conclude. Thank you for listening. I hope you found the content of this webinar interesting and relevant for your scientific work, and I would like to thank you for your attendance.
Thank you to both of you for that informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of your presentation window, type your questions into the box that appear on the screen, and click that send button. We'll answer as many as we can during the presentation. So let's look at our first questions from our audience members. We have quite a wonderful audience today. Our first question comes in from, what does the temperature inside a multifunctional plate reader depend on?
Well, I think I'm going to answer this. Well, primarily it depends on the reader design. The main heat sources are all types of electronic components, the power supply inside the reader, the light sources, so for example the flash lamp or the laser, whatever light source is used in a particular reader. And the longer these components are switched on and running, the more heat will build up inside the reader. So that is sort of the main contributor to instrument warming. It also somewhat depends on how old the instrument is, because over time dust can accumulate inside the instrument and prevent efficient ventilation. So a very old instrument will probably show higher temperature-related results variations than a brand new and modern reader. In addition, modern readers such as the Spark will have encapsulated measurement chambers that are better protected from ambient factors like light, temperature, and of course contamination. So they will be better suited for temperature-sensitive assays.
Wonderful. Thank you, Dr. Flatscher. Our next question is, you mentioned pre-warming as a surrogate for cooling the reading chamber, but doesn't pre-warming increase the reaction rate of the enzyme by prolonging incubation?
This is Gediminas Vidugiris. I believe this question to me more related. You're right, plate pre-warming will increase the reaction rate and will not reflect the reaction behavior expected at the room temperature, but the real purpose of a plate pre-warming is to achieve more even, less dispersed data and statistically better results. It is more important during the screening campaigns. It allows smaller signal variability, and it allows identification of the more hit compounds.
Wonderful, thank you. And I want to thank our audience members today. We have some wonderful questions coming in, and we appreciate your participation. This next question is for Dr. Flatscher. Which assays are most susceptible in temperature changes?
It's probably more related to me again question. I would say that all chemical reactions are equally susceptible to the temperature change, but what we observe is that a larger effect we see in multi-step bioluminescence assays, where probably accumulation of an active reaction intermediate takes place, and this is cause the larger signal increase during the plate warming.
Thank you so much. We have time for one more question from our audience. This one is directed actually to Dr. Flatscher. I already have a Spark in my lab, but without T-Cool, can I upgrade?
Yes, you can. However, it's a factory-based upgrade, so the instrument has to be sent back to be upgraded with the cooling module, so with the T-Cool module. I would recommend you contact your local sales rep for more information on how to do the upgrade, and I'm sure they will be glad to help you out on this.
Wonderful. Dr. Vidugiris and Dr. Flatscher, we appreciate your presentation today. Do you have any closing remarks you'd like to share with the audience before we close today?
I would like to take the opportunity to thank everybody for listening in. It was so exciting for us to share the results of our cooperation, and I'm really grateful for everybody who listened in today. Thank you.
And from Promega, we were happy to use the T-Cool module Spark reader to work with our assays, which helped, to achieve a better results. Thank you.
Thank you. Thank you both, and thank our audience members. I'd also like to thank LabRoots and Tecan, our sponsor today, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through April 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thanks so much for joining us. We hope to see you again soon. Bye-bye.